So good morning, everyone. So I'm Manoel Neres, and today I'm here to present the finding from my research. The title of my work is Microorganism-host interaction and the immunomodulatory mechanism of synthetic genome Mycoplasma mycoides species capri strain in capri peripheral blood mononuclear cell culture. This work has been supervised by Professor Dr. Marx from the Federal University of Bahia, Brazil, in collaboration with Brazilian agricultural research corporations and John Clay Venture Institute. So, Mycoplasma mycoid posters in the class molecules include bacteria that can cause disease in man ruminants, primarily goat, cow, and sheep. So it leaving significant economic loss in livestock. Up to the present moment, the clusters contains 5 species, with 2 deserving special attention caused the severity of their associated disease. Mycoplasma mycoides species mycoides, which can cause contagious bovine bird pneumonia, and Mycoplasma mycoides species capri, responsible for MAPS syndrome. So these clusters share the general characteristics of molecules, include free egg-shaped column and a reduced genome. Researchers have been often questioning whether it's possible to create a cell with a synthetic genome that is minimal enough to allow for the determinations of each gene's function. In 2010, Gibbs and his team started exploring these questions by chemically synthesizing the Mycoplasma mycoid genome from digital information and implant it into the cytoplasm of another organism. The first strain with a synthetic genome was named by Gibbs and his team JCVI1. Here are some characteristics of the JCVI1 strain. The cells are self-replicating and capable of logarithmic growth. Cells are small and ovoid. The rate of appearances of colony on other plates and the colony morphology are similar to the natural stream. And the synthetic genome with 1079 kilobases pair, also similar to the natural stream. In 2016, Huxon and his team reduced the JCVI1 genome to 531 kilobases pair. This process resulted in JCVI Syn3. Among the characteristics of JCVI3, we can highlight the presence of 149 genes of unknown functions, and the initial JCVI3 strain was extremely fragile and polymorphic, including extensive filamentations and vesicle formation during growth. To resolve the morphological distortions of JCVI3 in relation to the JCVI1 was incorporating 19 additional genes of JCVI1 that were not present in the strain. It resulted in JCVI3A with a phenotype similar to the JCVI1. So the next strain was JCVI3B. The genome is about 531 kilobases per and include a dual LOX P-late platform. This platform allows the inclusion of another genes. So we have studied the Mycoplasma-host interactions by evaluating aspects of immunomodulation in silic, in vitro, and the animal model. With this, this study aiming to assess the microorganism-host interactions and the immunomodulation mechanism of synthetic genome Mycoplasma mycoides species capri strain in capri peripheral blood mononuclear cell culture. For this, goat's blood was collected. After that, the PBMC was separated by fecal gradient, and of the PBMC extractions, the cell culture was infected with neomycin genome strain and wide strain. We have been using all of these all of these strains that we are talking about. So, clinical examination biochemical test of animal serum confirming that the animal were in good health, how you can see it here in this table. After cultivation, the supernatant was subjected to the alarmable blood test to evaluating the inhibitions of cell proliferation. Except for the 12 hours time point for DCVI3B, all synthetic streams are all evaluating times compared to the negative control. The wide stream inhibits cell proliferation when compared to the synthetic stream at all time points. After that, we realizing the ELISA test and dosaging of nitric oxide. In ELISA test, we evaluating all these cytokines, IL-1 beta, IL-4, and TNF-alpha. All tested stream could increase nitric oxide production at all analyzing infection time points, like we see here. And in ELISA test, IL-1 beta analyzing showed that all stream induced the productions of IL-1 beta after 6 and 12 hours. At 18 and 24 hours time points, only JCVI3A didn't cause an increase in the cytokine, how you can see in the graph. So to TNF-alpha productions, the wide stream induced significant TNF-alpha production at 6 hours and except for DCVI3B in 24 hours compared with synthetic stream and negative control. After 12 hours, JCVI3A and JCVI3B also induced TNF-alpha production. So at 18 hours, all treatment with synthetic stream is stimulating the productions of this cytokine. So when we are evaluating IL-4 productions for 6 to 18 hours, there is no statistically significant productions of the cytokine. After 24 hours, only for JCVI3A stream was there no significant increase. So this study demonstrated synthetic Mycoplasma mycoid capri stream potential in modulating the host pathogen interactions and immune response in cell culture. They inhibit PBMC proliferation and induce pro-inflammatory cytokine productions. Further studies include animal model and transcriptome analysis are crucial for understand this mechanism and develop therapeutic strategies against Mycoplasma mycoid capri and related pathogen. So I would like to thank all these partners and founding institutions, and I'm also very grateful to everyone from Micro Project at Federal University of Bahia, Brazil. Thank you so much.
Thank you, Manoel. Questions for Manoel?
I've got one. Hello, Manoel. Congratulations. Very nice presentation and very interesting results. What do you expect in the next stage of animal evaluation?
Well, the next step, we are realizing animal model and the transcriptome analysis. We believe that these analyses are crucial for a deep understanding of these previous results. So in the cell model, at first, the interactions is laboratory based and it's mimic natural infection. This means that other environment factor might influence immune modulation. Additionally, some genes with no function can play a role in the immune response. These are theories that we plan to investigate in the next step.
Thank you, Manoel.
Thank you.
Elívio, in our results, we are observing the GCVI3A is more related to modulate negative modulate of the response. Maybe this strain could be used for another model for the of vaccines, because the pattern of the response of this bacteria is not related with the other synthetic or wild bacteria. Maybe this bacteria we can use for this object.
Thank you,
I think
Lucas.
in animal model we can observe better the situation.
Sure. Thank you very much. Very interesting
Thank
results.
you. Thank you.
Were the differences between wild-type Mycoplasma mycoides and SIN1, would you call those significant?
We didn't observe the difference between the both strands related to cell model. Maybe in the animal model we can see the difference between both strands.